In this video, we will discuss about the effect of cytokines on the CAR-NK signaling and some of the ideas about CAR-NK therapy against the solid tumors, or what are the current research going on in different kind of solid tumors in terms of this CAR-NK therapy. So we will first start with the cytokine effects on the CAR-NK cells. In the previous video, we have discussed about effect of the receptors. Here we will discuss about the effect of the cytokines that are usually released by the tumor cells on the CAR-NK cells. So in the solid tumors are surrounded by tumor microenvironment, and tumor microenvironment most of the times contains this TGF-beta, and TGF-beta can interact with the TGF-beta receptors, either TGF-beta 1 or 2 that are expressed on the CAR-NK cells, and it might trigger different signaling pathways. One of them could be it will increase the phosphorylation of the SMAD2 and SMAD3, and this complex will then go to the nucleus where it will interact with the SMAD4 and thereby suppress the cytotoxicity effect by CAR-NK cells against the tumors. And similar kind of things are done by this reducing the mTOR and Siamese formations, and thereby it will suppress this CXCR3 and CXCR4, and that will inhibit the glycosylases. And the other things that can be they not suppress this mTOR Siamese, they overexpress this, and this will prevent the CXCR3 and CXCR4 to go into the nucleus, or they will suppress this CXCR3 and CXCR4 and also this CXCR1, and all this together will suppression of this will cause a decrease in the glycosylases. The other things that are triggered by TGF-beta receptors is this upregulation of the SOD, which will cause the reactive oxygen species formations within the CAR-NK cells, and that will also lead to the reductions in the cytotoxicity. And so these things are done by the TGF-beta that is not soluble. The TGF-beta that is soluble can directly interact with the receptors that are expressed on the CAR-NK cells, and thereby suppressing the activity of the CAR-NK, and which results in the reduced cytotoxicity. And there are some cytokines that can help in the upregulations of this CAR-NK efficiency or CAR-NK activations, and those are this interleukin 2 and interleukin 15, and this will forms a complex, this gamma C, and it can interact with this IL2 receptors beta, that will lead to the upregulation of this JAK1, JAK3 or STAT1, STAT5 signaling pathways, and that will increase the cytotoxicity and glycosylases. And similar things are done by when the interleukin 12 interact with this interleukin 12 receptors beta. It will lead to the upregulations of these JAK2 signaling pathways that will cause the upregulations of the STAT4, and that will also helps in the increased cytotoxicity and glycosylases. And this is one of the reasons why, while culturing these NK92 cells, we need this IL2 to activate the CAR-NK cells, or to keep the CAR-NK cells in the activation stage. Otherwise, these cells will die because they will have no activations, and this the NK92 cells needs IL2 continuously in order to keep them in the activation stage. And now we will discuss the figure on the right hand side, where some of the targets for different solid tumors are mentioned, and the research area has been going on those targets. We will start with this renal cancer. So in this case, the target is about this EGFR growth receptors that are expressed on these cancers, and the research is currently going on in the in vitro and in vivo at both stages. And for this glioblastoma, targets that are been identified using research is ERBV2, EGFR, B7H3, CD73, and so far this were positive results in the in vitro studies, and research is currently going on at that stage only. And for this lung cancer, the targets that are prominent targets that are identified is CCCR, DLL3, BA7H6, H3, and CD73, and both of them shows promising results both in the in vitro and in vivo studies. And currently the research is going on these 4 targets. For the breast cancer is EPCAM, HER2, EGFR, IICON, TF-CAR, NK cells, and these are the prominent targets. And uh, two of them shows promising results in the in vitro studies, and one of them shows EGFR shows the promising results in both in vitro and in vivo studies. No information about the other tools has been mentioned in the figure. The other one is this gastric cancer. It also has the 4 target genes, and out of which 2 of them, that is CEA and EPCAM, are being validated in vitro, whereas and whereas MSLN, CD19, MSLN or CD19 or NKGD2 are being validated both in vitro and in vivo. For the liver cancer, the common genes that are being that shows promising results for targeting this liver cancer is this CMAT, and it has been tested in vitro and shows the promising results. And currently they are working on, research is going on in this GPC3 CAR NK cells or L3C73 FC, L3C73 FC, that enhances GPC3 CAR NK cells functions. The last one that is mentioned in this figure is about pancreatic cancers. 3 main targets that is focuses on is MSLL, PSCA, FR alpha, and out of this FR alpha shows promising results in the in vitro, whereas PSCA shows promising results both in vitro and in vivo. And this CGAMP that is used in the pancreatic cancer cells can enhance MSLN CAR NK cells efficiencies. So if these 2 things are combined, then it will have a better results against diagnosis of the pancreatic cancers. So I hope you understood something about the effect of cytokines on the CAR NK cell signalings and some of the CAR NK therapeutic areas against the solid tumors, and what are the targets that are currently being hotspot in their respective cancer. That's all for this video. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. Thank you for your time.